हेलो और स्वागत है आप सभी का एक बार फिर से कोड टू सर्व अकेडमी में गाइस। और आज हम लोग कंप्लीट करने जा रहे हैं अपना बायोलॉजी का चैप्टर ट्रांसपीरेशन फॉर क्लास 8 जो हमारा आधे में छूटा हुआ था भाई। फर्स्ट लेक्चर हमने रिकॉर्ड कर दिया था, लाइव भी गए थे हम लोग, लेकिन सेकंड लेक्चर में ड्यू टू सम टेक्निकल इश्यूज उसको बार बार रिकॉर्ड करने के बाद भी कुछ ना कुछ गड़बड़ हो ही रही थी, तो फाइनली आज उस अधूरे लेक्चर को हम लोग कंप्लीट कर रहे हैं। फर्स्ट लेक्चर ऑलरेडी प्लेलिस्ट में पड़ी हुई है, आप जाकर देख सकते हो। आज के इस लेक्चर में वी आर गोइंग टू डिस्कस द सेकंड पार्ट ऑफ द ट्रांसपीरेशन। ठीक है भाई, शुरुआत कहां से करेंगे? आज के लेक्चर में वी विल स्टार्ट फ्रॉम द सिग्निफिकेंस ऑफ ट्रांसपीरेशन। इससे पीछे के टॉपिक्स तक हमने जो है वो बातें कर ली थी। आज हम लोग ट्रांसपीरेशन से के सिग्निफिकेंस यानी कि ट्रांसपीरेशन का क्या महत्व है, क्या इंपॉर्टेंस है, उस चीज को डिस्कस करने से स्टार्ट करेंगे। अब थोड़ा सा रिकैप दे दूं क्योंकि काफी टाइम हो गया है हमें ट्रांसपीरेशन पढ़े हुए, तो मैं कहूंगा ट्रांसपीरेशन क्या है? लॉस ऑफ excess water from the surface of leaf, from the surface of leaves to maintain the transport of वाटर। एक इंपॉर्टेंस तो हमने यही लिख दिया। ट्रांसपीरेशन है क्या भाई? ट्रांसपीरेशन एक ऐसा प्रोसेस है जिसमें प्लांट अपने एक्सेस वाटर को लूज करता है थ्रू इट्स लीफ सरफेस। और आज इसी ट्रांसपीरेशन का क्या सिग्निफिकेंस है, क्या इंपॉर्टेंस है, वो देखने वाले हैं। फर्स्ट ऑफ ऑल पहला सिग्निफिकेंस ये है भाई कि ये एक सक्शन फोर्स क्रिएट करता है। सोचो, यहाँ नीचे जो वाटर है ये नीचे से ऊपर की ओर ट्रेवल कर रहा है, ट्रेवल करके पहुंच जाता है इनटू द लीफ। ठीक है भाई, ट्रैवल करके कहां पहुंच जाता है? इनटू द लीफ। लेकिन ये हो किसके वजह से रहा है? ऑस्मोसिस के वजह से। ऐसा क्यों हो रहा है? क्योंकि रूट के पास कंसंट्रेशन हाई है और लीफ के पास वाटर का कंसंट्रेशन यूजुअली लो होता है, जिसके वजह से वाटर हायर कंसंट्रेशन ऑफ वाटर टू लोअर कंसंट्रेशन ऑफ वाटर के तरफ ट्रैवल करने लगता है। ठीक है, अच्छा, लेकिन क्या होगा अगर ये ट्रांसपोर्ट कंटिन्यूअसली चलता रहे? तो देयर विल बी अ सम पॉइंट ऑफ टाइम वेयर वी विल सी दैट द where we will see that the rate that the concentration of water here in the leaf will become equal to the concentration of water in the roots. What will happen next? The gradient, the change in concentration which was leading to the transport of the water in the whole process will stop. But does it stop? Does it stop during the transportation? क्या ऐसा होता है कि सचमुच में वाटर का फ्लो जो है वो लीफ के तरफ जाते हुए रुक जाता है? नो, इट डजंट। क्या रीजन है इसके पीछे? रीजन ये है कि लीफ डजंट होल्ड ऑल द वाटर विच वॉज बीइंग ट्रांसपोर्टेड टू इट। मोस्ट ऑफ द वाटर दैट रीचेस द लीफ, most of the water that reach the leaves escapes from there through a process called transpiration. अब अगर ट्रांसपीरेशन के वजह से ज्यादातर वाटर जो है वो लीव्स को छोड़कर निकल जाएगा, तो there will always be a maintenance of low concentration of water inside the leaf, which will lead to the continuous supply of water from the roots to the leaves. समझ आ रही है बात? यानी कि ये जो कंसंट्रेशन का डिफरेंस है ये बना रहे, इसके वजह से ये एक्सेस वाटर को क्या कर रहा है? लूज कर दे रहा है फ्रॉम द लीफ सरफेस, जिससे एक लो कंसंट्रेशन हमेशा यहाँ बना रह रहा है, बरकरार रह रहा है, और उसके वजह से वाटर हमेशा नीचे से ऊपर जा रहा है लीव्स के तरफ। ठीक है, अब ऐसा होने के वजह से यानी ये जो ट्रांसपीरेशन के वजह से ये जो डिफरेंस क्रिएट होता है इन कंसंट्रेशन, दिस क्रिएट्स अ फोर्स कॉल्ड ट्रांसपीरेशनल पुल। और ये ट्रांसपीरेशनल पुल क्या है? एक तरीके का सक्शन फोर्स है विच इज एक्चुअली सकिंग अप द वाटर फ्रॉम द रूट्स टू द लीव्स। इज दैट क्लियर? वेरी गुड, यही हम लोग फर्स्ट पॉइंट पे बात कर रहे थे। सेकंड, जस्ट लाइक आवर स्वेटिंग प्रोसेस, ये जो प्रोसेस प्लांट्स में हो रहा है, इट इज काइंड ऑफ अ वे टू कूल डाउन द प्लांट ऑल्सो। ओके, वी हैव अ व्यूअर हियर। सो मैं क्या कह रहा था भाई कि ये जो लॉस ऑफ वाटर है फ्रॉम द लीफ, दिस लीड्स टू द कूलिंग अप ऑफ द लीफ सरफेस। Just try to think that leaf is being there all the time, whole day, just absorbing sunlight. So it must have absorbed a lot of heat energy, due to which the leaf can become hot. And due to the raising of the temperature of leaf, the processes going inside the leaf will get affected. As we know, the photosynthesis is also a kind of metabolic activity, and metabolic activities do get affected by the change of temperature. तो ये चेंज का ये टेम्परेचर का चेंज ना आए, दैट इज वाई ये ट्रांसपीरेशन लीफ को ठंडा करने में भी मदद करता है. एज सून एज द वाटर लीव्स द लीफ, as soon as the water leaves the leaf, it actually cools off the surface of leaf, it cools off the surface of leaf and thus causing a cooling effect. ठीक है भाई. Now what is distribution of water and mineral, यानी कि ये ना distribution of water and mineral में भी help करता है. So how can we understand it? Right from the beginning, from where the water was getting absorbed by the roots and then being transported to the leaves, throughout the process it was carrying other substances too while moving inside the plant. When it had moved inside the roots, it also brought some minerals, some nutrients which was required by the plant, which seeped inside the plant along with the water. And when that water gets transported from the root to the leaves, it also takes those substances to the leaves also, to every part from where the water flows through the transportation process. Understood? And why is this flow keep going? This flow is keep going just because of the transpirational pull which is acting over here due to transpiration, and hence transpiration also helps in the movement of water along with the minerals. Okay, चलो भाई ये भी समझ में आ रहा आपको. Now let us move ahead, and now we will discuss how does transpiration affect the climate. Yes, you have heard it right. Transpiration also affects the climate. But how transpiration can affect the climate? So just try to think that there are a lot of trees in a place, and every tree is acting like a suction pump which is sucking groundwater and releasing most of it into the air in the form of vapor. Now these vapors will not remain there. Those water vapors will travel upwards to the layer where clouds are formed, and the transpiration in plants also leads to cloud formation, which leads to a better rain condition. So most of the plants on daily basis uses liter of water that is released in the atmosphere. This released water increases moisture in the atmosphere that brings rain. So this was the point I was trying to make out here in front of you, that all the water which the plant sucks up during the process of transportation, it loses most of it in the form of vapor into the atmosphere, and that water and those water vapors which got released by the plant actually gets converted into rain and causing better chances of rain, causing better quality of rain over the time. And that is why you must have heard that when we deforest an area in a large scale, it eventually results in the desertification of that particular land area due to the lack of rain clouds which was being formed when the trees were there and pulling out water from the ground and pushing it up to the layers of clouds. Okay, is that clear? Very good. Now let us move ahead. What is the relationship between transpiration and the photosynthesis? Are they related? Is the rate of photosynthesis gets affected by the rate of transpiration, or are they unrelated? So let us look into the topic itself first, then I will try to explain it to you. So first we are seeing here, process of photosynthesis requires water, which is an eventual fact, that no photosynthesis will occur if there will not be any transportation of water, as plants do not have their own water. They bring it by the process of transportation itself, right? And transportation of water is going going out, just due to the reason that there is the presence of transpiration in the plant. That is why the transportation process of water is keep running. Okay, very good. Now xylem provides the water to the site of photosynthesis for the leaves, we know it. So now I have totally understood that the rate of transpiration will affect the rate of photosynthesis, as water is the raw material required for the photosynthesis process, and transpiration is the reason behind the transportation of water to the leaves. So I can see a direct relationship between the transpiration rate and the photosynthesis rate, as if the transpiration rate will increase, the transport of water will also increase, but the holding of water inside the plant will get decreased, and vice versa. When the rate of transpiration will slow down, the transportation of water in the inside the plant will slow down too, but the holding capacity of water of the plant will increase when the rate of transpiration will decrease. Okay, understood? Okay, very good. Now as the water evaporates, in order to maintain a thin film of water over the cells, water is pulled from the xylem to the leaves. अब आपको ये पता है भाई. Now we know that when water reaches when water reaches the leaves, to maintain a constant thin film, it requires more transportation of water, so that the cells in the mesophyll layer always get rich supply, or are rich around with water molecules. Okay, so due to the lower concentration of water in the atmosphere, water diffuses into the atmosphere to create a pool. We have already discussed this part. Now actively photosynthesizing plants need more water, and availability of water can be a limiting factor for photosynthesis. Now this is what I have already told you, that the rate of photosynthesis will depend on the rate of transpiration, as higher transpiration rate will bring more water to the leaves, causing the leaves to have a faster rate of photosynthesis. Okay, and if the rate of transpiration or transport of water gets minimized, it will eventually minimize the rate of photosynthesis too in the plant. Okay, now let us look ahead. Why is transpiration important? We have already looked into it. Creates transpirational pools for transport, supplies water for photosynthesis, we have already discussed this point too. Transports minerals from soil to all parts of a plant. Okay, cools down the surface of leaves due to evaporation of water. As we know, evaporation is a cooling process. Whenever any sort of liquid evaporates from a surface, it causes a cooling effect on the surface. And what are we seeing in the process of transpiration is that, if this is the leaf surface, and if we are observing that water is being released in the form of vapor, or in the form of vapor by the process of evaporation from the leaf surface, it will eventually cause a cooling effect upon the leaf. Have you understood? Okay, very good. Now this same process, transpiration, keeps the cells turgid, hence it helps in maintaining the shape of the cells in the plant. Now try to think once, that have you ever noticed that the leaves are exactly upright and very in a very turgid manner, or in a very solid state when we see the leaves during the day, but when we observe the plant or the leaves of the same plant during the night time, we observe that the shape, or the erect or upright position of the leaves gets slightly lower down. Actually it is called nyctinacity, which means sleeping action of the plant. But how does the plant sleeps? What actually happens? Actually the water supply, which was being constantly supplied throughout the day, as the temperature was high, there was a great amount of light available. Also, the photosynthesis was actively taking place inside the leaf, so an active transport of water was being observed during the daytime as the transpiration was constantly going on. But the point is here, during the night time, photosynthesis almost stops in most of the plants. Also, the amount of water being transported to the leaves reduces due to the reduction of transpiration, as during the night time stomatas gets closed up, and closed stomatas actually forces the plant to reduce the rate of transpiration which was going on in the leaves. And due to the same reason, the turgidity, the amount of water that the cells were holding, also gets reduced. So this is the basic reason that when we see or observe the cells of the plants during the night time, we usually observe them in a less turgid state, just due to the fact that they are not getting enough water that was required for maintaining their maximum turgidity. But during the daytime, the transpiration is actively being performed there, due to which the turgidity is maintained at its highest state in the leaf, in the cells of the leaves, and hence giving the upright or erect position of the leaves during the daytime. So have you understood? So this was the topic. Okay, very good. Now let us look into an example that if transpiration is occurring or taking place inside the plant through the leaf surfaces, is there any proof by which we can prove that yes, transpiration is being conducted in the plant? So we are about to look into an experiment through which we can easily prove that the plant continuously loses water from the surface of its leaves, and this process is actually known as transpiration. Now let us just look into the experiment. Here we have a potted plant. Here we have a potted plant which in my sight looks in very healthy condition. So we have got a healthy potted plant. Now in this healthy potted plant, we try to cover it out with the help of a plastic sheet. Now try to take this plastic sheet as transparent as possible, as it will make it easier to observe the result of this experiment. Okay, now when we completely seal up a plant using a plastic bag, using a transparent plastic bag, and leave it for a few hours, and after a few hours when we come back and observe the same system, we see that a lot of water droplets have been formed during these hours when we were not seeing. Now from where does this water comes from? These water droplets are a result of, yes, very good, these water droplets are a result of transpiration. These water droplets are result of transpiration which was going inside the plant. So by the means of this experiment, we can easily prove the transpiration phenomena going with the plant. Okay, now looking ahead, what do I see here is, after some time you will observe that, okay, this is another experiment where we will try to prove that, now after the previous experiment, many of you could wonder that, sir, the water droplets that we are seeing here can be a result of a random phenomena. How can you say that plant is producing these water droplets? Some, yes, many of you can ask this question, but what I will answer is, I will answer you with another set of experiment which will now accurately prove that the water droplets that we were seeing inside the plastic bag in our first experiment were the result of the transpiration by the plants. Now, what will we do here is we will need three jars, jar A, jar B and jar C. And for the setup, we will set the jar A only with a potted plant inside it and then seal up it with a glass jar. Okay, simple setup for jar A. For jar B, what we will do is we will place a healthy potted plant, cover it with a transparent glass jar, but before that we will place a paper of cobalt chloride, cobalt chloride, which actually turns blue to pink when it gets exposed with water. Okay, and for the third jar we will only set up a blue cobalt chloride paper and leave all these three jars in the same condition as we have set them up for a few hours. And after a few hours, when we will observe them again, what we will see is that glass A or jar A will have same amount of water droplets, or the same observation which we were seeing in the first experiment, where we were observing a lot of water droplets on the plastic sheet, and just like that case we will observe a lot of water droplets inside the transparent glass jar. But for the jar B, the observation will be all the water droplets which were formed eventually during the process of transpiration gets exposed to the cobalt chloride paper, causing it to change into pink color, which is a property of cobalt chloride paper. Whenever it gets exposed with moisture, it changes its color to pink from blue. Okay, and for the third jar, where we did not have any kind of we did not have any kind we did not had any kind of plant there, what result did we get? We got that the cobalt chloride paper remained blue throughout the time, which means here there was no water droplets or not any sort of moisture being formed. Now this experiment proves that the water droplets are only being observed when a plant is placed inside the glass jar. Also, we have verified it by placing a cobalt chloride paper which changes color when the plant produces water vapor by the process of transpiration. And at the end, we saw that when we placed only a blue cobalt paper without any plant inside the glass jar, there was no change in color. So what does this experiment prove? This experiment proves that yes, the water droplets that we were seeing in the experiment 1 was the result of transpiration which was going inside the plant. Now, if transpiration is taking place in plants, is every plant transpirating at same rate? Is every plant transpirating the same amount of water? The answer is no. And if the answer is no, then how will we decide that how much water is being transpirated by any plant? Is there any method to measure the amount of transpiration occurring through a plant? Yes, there is a method, or I will say there are some methods. So the first method is the easiest one, where we use weighing as a method. Now, how can we use weighing as method? So for this experiment, we again need a healthy potted plant. So here we have a healthy potted plant. We'll pour some water inside the pot, and once the water stops draining from the pot, we will measure its weight, and after we measure the weight, we will set a transparent airtight pouch at the soil layer, so that no water no water could escape here by the process of evaporation from the soil itself. Okay, Now after some time, when we will wait again, after some time when we will weigh the same setup again, what we will observe that the weight 1, the weight 1 that we were getting here is far more than the weight 2 that we will get in the second observation. Now remember, we have already sealed up the pot with the help of a plastic sheet. It is already airtight, so no water could have escaped from this layer, from the soil layer. So how does the weight changes over time? There is only one option, and that option is the plant must have lost water through any another process. And what is that process? That process is transpiration itself. Understood? That process is transpiration itself. So the change in the weight, when we measured weight 1 and weight 2, the change in the weight is the amount of water transpirated during the whole process, or during a delta time. Basically delta time means during a span of time, the amount of water that was lost during due to transpiration is equal to the change of weight of the system. What is the system over here? The system over here is the pot and the plant itself, where the pot is sealed enough so that no water escapes from the plant itself, and water only being lost through the surface of leaves by the help of transpiration. So the change in weight we are observing is due to the loss of water by only the process of transpiration through the time period for which we were observing. Is that clear? Okay, very good. Now this is basically the initial weight. This is the same setup that we were trying to have back there, in which we have weighed the whole system of plant along with the pot initially, before the transpiration, and after transpiration when we try to measure the weight, we got the weight at as y kg, and before transpiration it was x kg, and we are very certain that x will be more then y, means initial weight of the plant along with the pot having more water due to having the initial transpiration phase will have higher weight, and lower weight will be of the final phase of the same system, where an amount of water lost into the atmosphere by the process of transpiration. And the change in weight, between the initial and final movement is basically amount of water transpirated. Is that clear? Okay, very good. Now moving up to the next, here we have one more method for observing the amount of transpirated water, or to observe the rate of transpiration going through a particular part of the plant. For this we will need a dissection or a dissected part of stem. Here we have taken a dissected or stem cutting of a plant, which we have dipped into the layer of into the layer of water. But what we have also done here is we have also put a layer of oil above the layer of water, which actually will keep floating above the water layer. And when we observe it for some time, we will see that the layer of oil will start getting down to the will start moving down. And what does this suggest? This suggests that the water is being lost in the form of vapors by the help of a process called transpiration, and the level of change which we observed from the initial phase to the final phase is actually the amount of transpirated water. What we can do is we can also calculate the change in weight of the previous system and later system. we can also calculate the volume of the water which changed from the initial condition to the final condition, and by every mean what we will get is the value of transpirated water. Okay, is that clear? Okay, very good. Now moving next, we have a device called potometer. What is a potometer? So in the potometer method, we use a device called potometer. Potometer is a device which is used to measure water intake by a plant. This intake is equal to the water lost through the transpiration. Basically, how much the plant will intake water will almost be equal to the amount of water it will transpirate. As we know that more than 95% of the water which plants takes up releases away, actually it loses away by the process of transpiration. Okay, now let us look into an example so that we can understand the working of a potometer. So potometer is a very simple setup where a plant, again a cutting, a stem cutting of a plant is placed in a water container which is attached, which is a part of basically the potometer, and this water container is attached to a tiny tube, a thin tube, where which is called as capillary tube, and this capillary tube consists of a measuring scale which is a volume scale, which actually tells the change in volume. How does it how does it tell the change in volume? Let me explain you. So here you will see that there is a formation of air bubble. We must introduce a, we must introduce an air bubble inside the potometer, inside the capillary tube of the potometer. Now when the water is getting absorbed by the plant, what it does to the water bubble is what it does to the air bubble is that the air bubble starts moving in the direction of transport 2, and the magnitude of the displacement that occurs in the water bubble marks the change in volume by the change in initial and final position. Basically, the difference between the initial and final position of the air bubble through the volume scale of the capillary tube is the volume of water which was transpirated by the plant in the process of transpiration. So this is what a this is what a photometer looks like, and this is how it works. Now there are some limitations basically to the use of photometer. What are the limitations? First of all, introducing the bubble itself is kind of a hefty task to perform during the use of photometer. First is that, and for the second reason we have, if we take a stem cutting for the whole experiment purpose, the survival of that stem cutting or the duration for which it will remain viable is a matter of variation. Like different plants act differently, and some stem cuttings could survive for a few days to some stem cuttings not surviving for even few minutes. Is that clear? Okay, very good. And also, as the measurement is being taken by the help of the movement of air bubble, what we will observe is that by changing the outside temperature, it will also affect the position of air bubble. So for the whole experiment of potometer, or for conducting any sort of experiment through the potometer, we will have to maintain a static a static temperature throughout the whole process, and that is the very limitation. So we have also seen three limitations which are associated with the use of photometer. So everyone, this was the remaining part of the chapter transpiration. I hope you all must have got a very great idea regarding the application of the transpiration, that how it helps the plant, also what is the importance, how it is linked with the climate, and also how we can measure the rate of transpiration or the amount of water which got transpirated during any sort of transpiration span. So this was all for this today's lecture, and I promise to meet you in another lecture where we will talk about some another fun topic and some another chapter. Till then, bye bye.